This recorded session is dedicated to the continuation of our lecture on biosynthetic pathways for metabolic production in microbial organisms. As mentioned, we divide, or this module is actually divided into two parts. So, part one, we already started the on site discussion last February 22. So, I'll be recording two sessions to complete part one. And then next week we will be completing, or I'll have a separate lecture on part two. So, as a review of what we have discussed so far, we looked into the following concepts. We reviewed microbial metabolism in general, the importance of um, sugars as primary or central substrates in the production of different types of metabolites and the associated activities such as energy release, how it happens. Also reviewed fermentation um, in the context of microbial biosynthetic activity. We defined the difference between fermentation and respiration. Also, we reviewed ATP generation as a parallel activity to um, substrate metabolism and what happens in general, the concept um, of uh, chemiosmotic ATP generation in microbial systems. So we already introduced fermentation, then the different glycolytic pathways that bacteria make use of, and the EMP as the classical primary pathway. Then we also discuss on the concept of natural product biosynthesis, its evolution, or the idea that microbes are able to produce bioproducts that are usable, no, to humans. Okay. We define the differences between primary and specialized metabolites in terms of when they are produced. We acknowledge that in reality, specialized metabolites are which classically have been identified as secondary metabolites because they are produced when the bacterial culture achieves a stationary phase. So we redefined it, or we accepted the fact that in reality, in environmental settings, in non-laboratory settings, they're not microbes exist as communities. So their secondary metabolites are not necessarily always produced during the stationary phase, since the bacterial growth curve, as we know it, is actually more applicable in a laboratory setup. And essentially, those secondary metabolites or special, or as we proceed with our discussion, those specialized metabolites may be produced or may be produced at a different timeline, or following a different timeline, not entirely following the laboratory timeline, or the growth curve of microbes as we see in the laboratory. And given that there are a mix of interactions in any given specific environment, we look into the different factors affecting microbial bioproduction. Okay, so we define the difference between primary and specialized metabolisms. Also, the key examples of molecules that are classified in each bin, And then we introduced the concept of the parvome, which are all the small bioactive compounds associated with microbial systems. We also talked about some examples of questions relating to the biology or the general biology of the parvome, and these questions led us to the deduction that parvome components could be a potential new component of the central dogma of molecular biology, the process of information work, the information transfer from one molecule to another is actually it is actually also influenced by by small bioactive compounds produced by microbial living systems. And we acknowledge that there is a gap, there's a black box with regards to how we shifted our view in appreciating low molecular weight compounds within the parvome of a bacterial species or a microbial species. Um, and how these low molecular weight products have been utilized for human activity. Okay. We also talked about the importance of prioritization in natural product discovery, as we acknowledge that there are a lot of potential sources of parvo molecules. So there's the issue or challenging question on which microbe or product to prioritize in terms of downstream analysis and upscaling. Last time, we already introduced different primary key examples of primary metabolites, which are universally needed molecules for microbial organisms to start surviving or to continue surviving in a given environment. And these primary metabolites, we listed a few of them. Um, and in this recorded lecture, we will emphasize on the different pathways that are involved in the production of these key primary metabolites. So first, let's have a look into the 13 precursor metabolites for macromolecule building block biosynthesis. So there are 13 central metabolites which are shared by different pathways or which can be produced and found in different biosynthetic pathways. So these 13 precursor metabolites um, are involved in the different primary biosynthetic pathways. Okay. So first, let's take a look at glycolysis. So glycolysis is the oxidation of sugar molecules in order to generate ATP, reductants, and pyruvate. Okay. Glycolysis uh, in different microbial species may have some specific differences in terms of the enzymes that are used in the different steps. You know? In general, the process is an amphibolic reaction. It's found in almost all organisms. Amphibolic meaning there's a catabolic component, there's a breaking down component, and there's an anabolic component, which is building up. No? And in living organisms, glycolysis is essential in all conditions of growth. So the input for glycolysis are sugars and other, particularly glucose and other six carbon sugars. That's why the term hexose is introduced. So glucose can enter the microbial cell via a phosphotransferase system and is converted to a phosphorylated form or intermediate G6P via the action of glucokinase. Okay. The output is a phosphorylated hexose, beta D glucose 6 phosphate. Okay, can also be a beta D fructofuranose 6 phosphate. Okay, and additional intermediates such as GP, glycerone phosphate, 3-phosphodiglycerate, PEP, phosphoenol pyruvate, and most importantly, pyruvate, which will enter succeeding pathways. So glycolysis has, as I mentioned, has multiple variants depending on the enzymes that are utilized in particular steps. So glycolysis 2 makes use of F6P as its primary substrate. Glycolysis 3 makes use of glucose uh, makes use of glucose as a primary substrate. Other variants of glycolysis also depend on the species. So, for example, glycolysis 5 is found in species under the genus Pyrococcus, and in glycolysis 6, fructose is the primary substrate. Okay, glycolysis is also synonymous with the EMP pathway, the Emden-Mayer-Parnas pathway. So here we have a visual representation of the glycolytic pathway. So at the once that the sugar molecule has been translocated inside the cytoplasm of the cell, so there are multiple steps that will catalyze its final conversion to pyruvate acid. Okay, and there are several enzymes that are involved. So from your common your glucose molecule can be broken, can be what the initial step is. It's phosphorylation inside the nucleus. No, I mean sorry, inside the cytoplasm. Okay. Wait. A few more things here. Okay. So I I mentioned about pyruvate. So pyruvate is the primary output of glycolytic pathways, and pyruvate enters a succeeding pathway, which is the TCA cycle, the tricarboxylic acid cycle. So TCA generates energy and reducing power, and it exists in all oxygen-dependent or aerobic organisms, very common in heterotrophic bacteria and archaea. May also happen in facultatively autotrophic archaea if they are grown in heterotrophic conditions. No? The primary input is acetyl-CoA, which is derived from pyruvate. So once pyruvate is converted to acetyl-CoA, then acetyl-CoA will enter TCA and will allow for the pathway to turn cycles. No? The output for TCA cycle include carbon dioxide, where two molecules of it are produced per turn. So one molecule of acetyl-CoA is used per cycle or per turn. And additionally, aside from CO2, other outputs of the TCA cycle include reduced molecules. No, so there's reduced NAD. So there's additionally we have NADP, ketones, and GTP. No? There are variations of the TCA cycle in microbes in prokaryotic microbes when compared to eukaryotic pathways. So first, NADP-dependent enzyme catalyzes the dehydrogenation of isocitrate to ketoglutarate, while in eukaryotic pathways, it's different. There's a NAD plus minus dependent enzyme that does the work, that does the step. Also, catalysis of malate to oxaloacetate, or the conversion of malate to oxaloacetate, is following or is dependent on a quinone-dependent enzyme when compared to to eukaryotic pathways where a NAD-dependent enzyme is used. Okay. So remember the the TCA cycle. So from acetyl CoA to the conversion, several conversion steps. So from citrate to isocitrate to oxoglutarate to succinate to fumarate, and then eventually to oxaloacetate. So take note, it is a cycle as acetyl CoA is actually converted or it's actually primarily um, uh, oxaloacetate as the uh, uh, end and start molecule that will turn the cycle, that will keep the cycle turning as available acetyl CoA molecules are present, no? Okay. Then we introduce different living organisms according to their metabolic diversity. So in relation to the primary biosynthetic pathways on energy production and sugar sugar breakdown, sugar oxidation. So we have microorganisms, phototrophic living organisms that are known to be able to absorb radiant energy, light energy, via specific pigments that will result in the excitation of electrons present in enzyme complexes, and that event will provide oxidation power, producing oxygen for the case of oxygenic photosynthesis, or not producing it at all in the case of non-oxygenic photosynthesis. Now, we also have living organisms that are dependent on oxygen, those that do aerobic respiration, where molecular oxygen is the final acceptor of the electrons. Then there is anaerobic respiration, where oxygen is not required. Usually, we expect anaerobic respiration in microbes that are optimized or that have been adapted to conditions where molecular oxygen is reduced, limited, or totally absent. No, so other inorganic ions such as nitrates, sulfates, and carbonates can serve as final electron acceptors for the case of anaerobic respiration. And there's the concept of fermentation, where an organic compound that is often a metabolic intermediate coming from the oxidation steps of another organic compound will serve as the terminal oxidant, no? And the resulting outputs are more reduced organic molecules. Okay. So we relate this to some examples of microbes, and there some examples of key microbes that carry out fermentative or respiratory processes, no? Or respiration processes. For the case of microaerophilic fermentative microbes such as lactic acid bacteria, there is no or very low concentrations of oxygen, usually 10% or below the atmospheric concentration. So remember, the atmospheric concentration of oxygen is about 20%. So microaerophiles do not really exist or cannot thrive in atmospheric concentrations of oxygen. And for lactic acid bacteria, there is the production of large amounts of lactic acid, which will drive pH decrease in the environment. No? Examples of this are fermented food products. Then we also have facultative anaerobic microorganisms such as yeasts, which thrive with or without oxygen. Um, and they are known to have preferential consumption or preferential activity or preferential high activity in environments with high sugar substrates. So that they are able to consume sugar, very high concentrations of sugar. Okay. For the case of enteric bacteria, no, those that carry out facultative anaerobic respiration can optimize the condition of the GIT of warm-blooded animals. So actually, enteric bacteria has co-evolved with warm-blooded animals, and their facultative anaerobic processes or their anaerobic respiration within the GIT has optimized the nature of the GIT over time. Okay. Then there is obligately anaerobic as well as other facultatively anaerobic microorganisms which we find in the environment and soil and water systems. So they carry out the conversion of organic substances under conditions without oxygen. So very good example of this is methanogenetic or methanogenesis based organisms. So the production of low molecular weight organic acids, alcohols, carbon dioxide, and hydrogen gas is associated with them. No? Then syntrophic microorganisms, which we relate under anaerobic respiration, facilitate the conversion of organic compounds to hydrocarbons, which is a key feature of methanogens. Okay. Now let's take a look at acid production. Okay, so mixed acid fermentation is when bacteria, when the specific microbe can use two or more pathways during the terminal steps of fermentation and will produce a mix of different organic acids. Okay. So, as a review, okay, from your organic substrate being broken down, okay, to your primary metabolites, okay, such as amino acids, other forms of smaller sugars, and organic acids, okay. Alongside with it, fermentation processes can is a route that is potentially that can be potentially used and will produce a number of different end products such as ethanol, alcohols, acids, lactic acid, glycerol, propionic acid, acetic acid, butyric acid, okay, as well as the production of hydrogen gas and carbon dioxide. So you see there's a diversity of output products from fermentative processes. Okay. Now, for mixed acid fermentation, it's characteristic of different genera of bacteria within the Enterobacteriaceae family. So groups such as Citrobacter, Escherichia, Proteus, Salmonella, Shigella, Yersinia, and Vibrio, and to some species of Aeromonas, they can carry out mixed acid fermentation. And it can also be carried out by anaerobic fungi. The final amount of end products will depend on the species, on the nature of the microorganism, and the growth conditions that are present during their during the carrying out of fermentative processes. Acid to neutral product ratio is actually 4 is to 1. So the environment is more acidic, 80% acidic than neutral. In bacteria that possess FHL complex, formate hydrogen lyase complex, there is equimolar production of carbon dioxide and hydrogen gas. Okay, so take note of this. In bacteria that express the FHL complex, there is equimolar production of carbon dioxide and hydrogen gas. Key enzymes for the case of fermentative processes of mixed acid fermentation actually control entry, okay, into mixed acid fermentation pathway in the absence of oxygen. So oxygen negatively regulates the key enzymes that allow a bacteria or a microbe to approach further breakdown of the substrate uh, following the mixed acid fermentation route. So there is a requirement for anaerobic conditions. So if there is very low concentration of oxygen or no oxygen at all, then entry into the mixed acid fermentation pathway is potential or is inevitable, as oxygen will down regulate or will in not necessarily inactivate but down regulate the activity of the key enzymes that allow the entry of a bacterial species into the mixed acid fermentation pathway. Okay. Then we have alcoholic fermentation. Okay. And in this figure, you can see different switch or two potential routes. So we have alcoholic fermentation. The end product is, as an example, is ethanol. And we have the glyceropyruvic pathway or the glyceropyruvic fermentation where end product is glycerol. Okay, so your microbe carrying out glycolysis of your glucose to your end products, no, may utilize any of the two pathways depending on the condition. Okay, since the start point intermediates, dihydroxyacetone for GPF and G3P, glyceraldehyde 3-phosphate for alcoholic fermentation. Are uh, they are reversible, or they can be intra-converted, no, to each other. So there is a potential for either two pathways to be activated depending on succeeding conditions. But in the case of fermentation, always remember there is a lack or very low concentration. There is a need for anoxic conditions or anaerobic conditions for fermentative processes to take place. Okay. So alcoholic fermentation has been known for the longest time to be involved in the transformation, stabilization, and conversion of very sugar-rich substrates, often carried out by yeasts and other fungi and bacteria. Okay, so there is the need for, or during alcoholic fermentation, formation, usually end product of the EMP pathway. Pyruvate is end product of glycolytic pathway, be converted to acetaldehyde via the action of pyruvate decarboxylase. So there's decarboxylation producing acetaldehyde. So going back to this figure, so you can see that after or once pyruvate is available, okay, in the presence of a decarboxylase enzyme, it's converted to acetaldehyde, and acetaldehyde can be further converted to ethanol. Okay. Um, I mentioned about its parallel existence with the GPF pathway. So it's associated with glyceropyruvic fermentation. In the past, GPF has been exploited for the production of glycerol, which is important during the 50s. No? The shift from alcoholic to GP fermentation is driven when there is a need to regenerate NAD+. And there are several potential reasons behind that. Could be that there is no acetaldehyde available, there is no or low activity of the decarboxylase enzyme of pyruvate decarboxylase, and there is already high rate of acetaldehyde conversion to acetate, especially if the microbial system is expressing aldehyde dehydrogenase, which catalyzes acetaldehyde to acetate conversion. Okay. So in mixed acid, in alcoholic fermentation, redox balance is achieved via NAD regeneration. And that happens when acetaldehyde is finally reduced to ethanol in the via the activity of alcohol dehydrogenase. The ATP yield for alcoholic fermentation is 1 or 2 moles per glucose molecule depending on the glycolytic pathway that was utilized. If it's EMP pathway, that's 1 ATP per glucose molecule. If it's the Entner-Duodorov, the ED pathway, there's 2 moles of ATP per glucose molecule that are produced. Okay. So in bacteria, a known species, a very industrially significant species for alcohol production, for alcohol fermentation, is Simomonas mobilis. Simomonas mobilis is a known bioethanol producer. So it thrives in very alcoholic conditions. It can convert 1 mole of glucose to 1.9 mole of ethanol, and it's a very rapid fermenter. It's even faster than Saccharomyces cerevisiae. Cellularly, Simomonas mobilis is gram-negative, it's facultatively anaerobic, and it's an ethanologenic bacterium. So it's really, its bioactivity is preferential to the presence of ethanol, or to the production of high concentrations, high amounts of ethanol. It's actually adapted and very tolerant in conditions of high ethanol concentration. So in industrial settings where of course the fermenter will keep on producing ethanol in its in allowing the product the when allowing the activity of this species, continuous production of the output does not inhibit the biology, the integrity of the microbe, since it can persist in such environment. They're very alcohol loving. Okay. Simomonas mobilis uses ED pathway to metabolize glucose, fructose, and sucrose, and usually, or in general, no, 1 mole of ATP is produced per mole of sugar. Okay. Then we have Saccharomyces cerevisiae, which is the most important microbe in alcoholic fermentation. So it's the one that the species that we've been exploiting for alcohol development, for alcohol bioproduction. And over time, okay, we have already domesticated Saccharomyces cerevisiae. It can be considered as a domesticated microbe. So meaning we already adapted it to industrial settings. Another another example, another pathway I'd like to introduce is propionic acid fermentation. Okay, so propionic acid fermentation is carried out by species in the genera Propionibacterium, which I think has been assigned a different genus name. Also, I think now it's called Cutibacterium, and also Clostridium, which is a soil inhabiting genus. The substrates that are used for during propionic acid fermentation is um, particular hexoses like glucose and lactate, no, and the pathways that are utilized, as well as the outputs in terms of carbon dioxide, ATP, and the end product differ based on the substrate that is used. If it's a hexose, 3 moles of hexose or sugar, Pyruvate production is via the EMP pathway. Okay, pyruvate to pyruvate carboxylation to oxaloacetate is carried out via the action of or via the presence of methylmalonyl coenzyme A, and there is reduction to propionate. Okay, 4 moles. If lactate is used as a substrate, okay, uh, instead of 4 moles of propionate or propionic acid, only 2 moles are produced. Additionally, it's associated with the liberation of 1 mole of acetic acid, 1 mole of carbon dioxide, and 1 mole of ATP. So lactate as a substrate is a lower yield pathway, no? Or it uses a lower yield pathway. Because compared to lactate, if a hexose has been used, if glucose has been used, acetic acid is produced at 2 moles, carbon dioxide at 2 moles, and more ATP is actually produced, 12 moles. Now we have lactic acid fermentation. So lactic acid fermentation is carried out by species of bacteria, the common Bifidobacterium, some species of bacillus, some protozoa can also carry out lactic acid fermentation, and some examples of water molds. In eukaryotes, in humans particularly, skeletal muscle cells carry out lactic acid fermentation. So when you work out your skeletal muscles, you actually produce lactic acid, and that's associated with the tiring of your muscles. Uh, Lactic acid fermentation can produce lactate. For the it can produce lactate, ethanol acetate, and carbon dioxide. No, so there are two pathways: homolactic and heterolactic. For homolactic fermentation, the output is lactate, while for heterolactic fermentation, there's a mix of outputs. There's lactate, ethanol acetate, and carbon dioxide. Okay, and in some cases, for if the bifid shunt is activated, lactate and acetate is happening, especially for the case of species thriving animal GIT. So they carry out the bifid shunt, and lactate and acetate are produced. So lactic acid fermentation is associated with the souring of milk products. Okay. So as a review, so take note of the following figure, the visual. So your glucose carrying out glycolytic process via glycolytic pathways will produce pyruvate. Pyruvate, when fermented, when entering the fermentation, when entering specific fermentation pathways, can produce lactate. And the production of lactate results to regeneration of NAD plus from NADH. Okay, let's examine homolactic and heterolactic acid fermentation. So homolactic acid fermentation is carried out by species under Lactococcus, Enterococcus, Streptococcus, Pediococcus, and some species of Lactobacillus. Homolactic acid fermentation, the last step is carried out by the enzyme LDH, lactate dehydrogenase, allowing for hydrogen transfer from NADH to pyruvate. Okay? And in homolactic acid fermentation, the transport of sugar is carried out either by a phosphotransferase system or a permease complex. So all sugars are activated to beta D fructofuranose 1,6 bisphosphate via different routes, depending on the nature of the sugar and on the way that it enters the cell. No? To discuss more about it, no? Intracellular fructose, which enters via a permease, is phosphorylated first to fructose 6 phosphate, then to fructose 1,6 bisphosphate. Okay. Sugars are metabolized via the EM pathway. We have glucose, pyruvate, and then lactate. Okay. 2 moles of ATP are produced per mole of oxidized glucose for the case of homolactic fermentation, homolactic acid fermentation. Okay. Then for heterolactate acid or heterolactic acid fermentation, species that carry out this pathway include those that are within the genera Leuconostoc, Enococcus, and Vizela, and some lactobacilli that are known to function in heterofermentative conditions. Okay. The absence of glycolysis is due to the lack of specific enzymes such as aldolase and isomerase. It does not carry out EM pathway, okay, unlike homolactic acid fermentation, and rather will make use of a phosphoketolase pathway for pyruvate formation. No? Sugar internalization can be carried out by a permease, which is non-phosphorylating, and sugar phosphorylation is carried out by a dedicated kinase. Okay. DG6P, which is produced after the sugar has been internalized by the microbial cell, will enter the PPK pathway and will result to the production of DG3P, the glyceraldehyde 3 phosphate, acetyl phosphate AP, and carbon dioxide. The final products are lactate, ethanol, and carbon dioxide. Acetate may also be produced if there's specific conversion enzyme available. And unlike homolactic fermentation, in heterolactic fermentation, only 1 mole of ATP per mole of glucose oxidized is produced. Remember, compared to homolactic, where 2 moles are produced, 2 moles of ATP are produced. In heterolactic acid fermentation, only 1 mole of ATP per mole of oxidized glucose is produced. Okay. So here we can see a side-by-side comparison of homolactic and heterolactic fermentations. So A is homolactic fermentation, so take note. So glucose, once it enters the cell, is phosphorylated to G6P. For the case of homolactic fermentation, there is conversion of glucose to fructose 6 phosphate and bisphosphate eventually. Then there's production of glyceraldehyde, 3, 3P glyceraldehyde, then pyruvate, it's further converted to pyruvate, and pyruvate is then converted to lactate. And take note of ATP production in homolactic fermentation. Okay, net of 2 ATP moles since ATP is consumed during sugar internalization. ATP is also produced during conversions of glyceraldehyde to pyruvate, as well as the phosphorylation of additional phosphorylation or secondary phosphorylation of F6P to bisphosphate. Okay. And then we have for heterolactic fermentation, so glucose once internalized becomes phosphorylated, produces 6P gluconate. Then we have conversion, additional conversion since there's no isomerase present. Okay, there are intermediates that are very different. So there is ribulose 5B and corresponding carbon dioxide liberation. There's cellulose 5B, and there is conversion to glyceraldehyde 3 phosphate and acetyl phosphate. Glyceraldehyde 3 phosphate follows the further conversion to pyruvate and lactate, and the usual regeneration of NADP and production of ATP, while the corresponding acetyl phosphate can yield ethanol. Okay. We also have butyric acid fermentation carried out by species under the genera Clostridium, which are anaerobic microbial system, like microbial species. They often occur in systems with pH that are not low enough for the exclusive activity of lactic acid bacteria. So the breakdown of the substrate, the sugar molecule, um, is also corresponding or parallel with some other types of uh, carbohydrates, some other types of carbohydrates such as pectin and amylose, if they are present. No? So key enzyme facilitating the conversion of pyruvate to acetyl-CoA is a pyruvate ferredoxin oxidoreductase. So take note of this enzyme for butyric acid fermentation. Pyruvate ferredoxin oxidoreductase. And the outputs are carbon dioxide, hydrogen gas, acetic acid associated with energy liberation, and butyryl CoA. So there are 3 moles of ATP that are produced during butyric acid fermentation using 1 mole of oxidized or glucose for oxidation. Okay. Okay. So now that we've covered a discussion on mixed acid fermentation and alcoholic fermentation, no, I want to emphasize that in nature there are different ecological niches and interactions among fermenting microorganisms. Okay, and in relation to some industrial applications as well as specific ambient conditions, um, let's have a summary. This visual is a good summary of that. So in cases of fermented milk products and sourdough production, your primary activators or species of concern are lactic acid bacteria and yeast. In the ripening or maturation of cheese products, propionic acid bacteria and lactic acid bacteria are the primary activators. In the GIT of GIT of warm-blooded vertebrates of animals, primary activators that carry out different fermentative pathways are lactic acid bacteria, Vipidobacterium, please remember this, Vipidobacterium, and enteric bacteria. And in the case of silages, lactic acid bacteria, Clostridium, and enteric bacteria are the primary activators. Okay? I'll divide the continuation of part 1 into 2. So this will be our endpoint slide for this recording.